I will call the remote hearing of the Environment and Natural Resources Finance and Policy Committee to order. Today is September 14th, 2021. This meeting is held in accordance with Rule 10.01, which was passed and allows for remote hearings. All remote hearings are recorded and live streamed by House Public Information. Members, we won't take any official action today, but we do need to take the roll. So staff, take the roll.
Chair Hansen is present. Vice Chair Wasilwick.
Wasilwick present.
Heinzelman Josh.
Heinzelman present.
Acom Patty.
Acom present.
Ackland Susan.
Ackland present.
Backer Jeff.
Backer present.
Becker Finn Jamie.
Becker Finn present.
Eklund Rob.
Present.
Fisher Peter.
Fisher present.
Green Steve. Green Steve. Igo Spencer.
Igo present.
Jordan Sydney.
Jordan present.
Keeler Heather.
Keeler present.
Lee Fu. Lee Fu. Lippert Saad.
Lippert present.
Representative Lewick is excused. Morrison Kelly. Morrison Kelly. Nelson Nathan.
Nelson present.
Tice Tama.
Vice present.
And just quick run back. Green Steve. Green Steve. Lee Fu. Lee Fu. Morrison Kelly. Morrison Kelly. A quorum is present.
Thank you. The quorum is present. Today we are going to have an update on chronic wasting disease. There was quite a bit of activity during the special session, and a lot has been happening with research. We want to hear how the agencies are working together. Our first speaker, we always tend to leave Dr. Larson towards the end, and we want to make sure he has enough time. So we're going to start with Dr. Peter Larson and his team. Because we have a packed agenda today and we want to make sure that we're getting the public testimony in, we've set up a timeline. So we've got Dr. Larson scheduled to finish about 10:25. We'll take questions after each presentation, but try to align within that timeline. So Dr. Larson, if you could introduce yourself for the record and begin.
My name is Dr. Peter Larson. I'm an assistant professor in the Department of Biomedical and Veterinary Sciences at the University of Minnesota. Chair Hansen and members of the committee, thank you for the opportunity to appear before you today. I'm very happy to share MinPro's latest outreach and research efforts with the committee. And just to, as way of a bit of background, MinPro, our research and outreach center, was established in 2019. This is an innovative R&D think tank with a growing portfolio of research efforts that I'll highlight later in the talk. I'm co-director of MinPro with Dr. Tiffany Wolf, and Mark Schwabenlander is associate director. And everything that I'll share with you today is a product of a large team, a growing team of over 70 MinPro research associates, staff, students across the United States, and that is growing month to month. We're very excited and proud about that. Just to outline of the talk today, I'll begin by highlighting some of the outreach efforts that our MinPro team has conducted over the last year, then a bit about some of the most recent research outputs with respect to RT-Quick, which I had talked about previously with this committee, but also our other prion diagnostic capabilities. And then I'll finish with a few slides on the ongoing forensics analysis of the Beltrami County carcass dump site, which has been in the news since April. And we'll be highlighting some of the results that we have generated from the site itself. For our outreach efforts, I shared with the committee and with the public this article that was published just last week. Mark Schwabenlander is first author, Dr. Roxanne Larson is senior author, this really highlights all the outreach activities that MinPro has done over the last 2 years. And if you read the introduction of this article, it mentions that it was in February of 2019 of appearing before this very committee that several members of the committee, including Representative Wagenius, were very animate that the University of Minnesota needed to do more on CWD outreach. And I can say that since that moment in time, we estimate at least 6,000 Minnesotans have been reached through our outreach efforts before the pandemic through one-on-one lectures, seminars. During the pandemic, we've had a transition to online activities. But we are doing as much as we can to connect with Minnesota's public on CWD. And we're also developing innovative educational tools. So this includes printing 3D, full-scale 3D white-tailed deer heads. And I'm showing you an image of one here. These are used to be able to help educate hunters and farmers on where to extract tissues that are commonly used for CWD testing. Down at the bottom here, I'm actually showing where we are lending these out to our DNR colleagues, both within Michigan, but also here in the state of Minnesota. Kelly Straka, who I believe is the DNR in Michigan, is going to be joining the DNR here in Minnesota. She requested using one of these so they can help train their staff and members of the public on how to collect tissues for CWD testing. So we're doing innovative things like that. But I encourage everyone to read the article that was published last week to get an idea of the full scope of our outreach efforts. We are also engaging with the Amish, Hmong, and tribal nation communities on CWD. There's been several visits to Amish community in spring of 2021, led by Dr. Wolf and Mark Schwabenlander. We conducted interviews to better understand community level knowledge and concerns of chronic wasting disease. And this is around the Harmony, Granger, and Canton areas in the southeastern portion of the state. For our Hmong and tribal nation community, we are conducting interviews to identify critical educational needs that can directly inform our outreach material. So it's very, very important to develop a deep understanding of cultural values and norms to really help custom tailor our outreach efforts to have a greater impact. So this is an ongoing effort, again, impacted by the pandemic, but the interviews have been ongoing via Zoom interface. And that work is led largely by Dr. Wolf and Mark Schwabenlander. We're also assisting tribal nations in the establishment of a CWD surveillance network. And this is, again, work by Dr. Tiffany Wolf to interact with as many tribal nations as we can, but also with the DNR to help foster and develop this CWD surveillance network, especially in light of what's happened in Beltrami County, which I'll talk about a little bit later. So moving on to our diagnostic R&D efforts. I've presented to the committee in the past about RT-Quick. So RT-Quick is this really sensitive and robust prion detection assay. The developer of RT-Quick has been quoted as saying that this tool, if you will, can detect CWD prions at the amount of a tablespoon in 400 Olympic swimming size pools. So it's very sensitive. This has become our workhorse, our primary CWD diagnostic workhorse in MinPro Lab. We have protocols for multiple tissues, blood, feces, soil, water, plants, even insects, which I'll talk a little bit about when we get into the forensics analysis of the Beltrami site, but also even surface swabbing. So this has a wide range, a wide utility to be able to detect prions in a variety of both biological and environmental settings. As far as we know, we are the first team to epidemiologically validate RT-Quick using Chair Hansen. Should I field questions now or wait till the end of the talk?
Wait till the end of the presentation.
Okay.
They'll line up. We'll see multiple hands.
Okay, roger that. So thanks to the DNR, the Board of Mental Health, and the USDA, we've been able to epidemiologically validate RT-Quick using tissues from both natural and farm populations. We need to expand that to include the same tissues that are used frequently for ELISA and IHC, and I can talk a little bit more about that probably during the question and answer session. But this is really important because what we've shown is that RT-QuIC is just as sensitive when using similar tissues that are used by ELISA and IHC. It's just as sensitive. And if you use tissues like, for example, tonsils, it seems like RT-QuIC does a better job at detecting CWD than ELISA or IHC, the traditional methods that are used for diagnostics. This RT-QuIC can detect prions in samples that traditional tests can't use. For example, decomposing tissues, feces, blood, environmental samples. There was a big push 2 years ago for a live animal test, and I have included in the packet to the committee our one-pager that describes how this method can be used as a live animal test. And I've talked and presented that information in front of the Board of Animal Health, and I believe to this committee in the past. So that option is available. It is a matter of how does the industry, the Board of Animal Health, and the state want to proceed with RT-QuIC as a live animal test. We've also recently published this paper just within the past few weeks that describes that we can detect CWD prions in white-tailed deer muscles, and this is through collaboration with the DNR. It is a common misnomer that the prions, that infectious prions that cause CWD are only found in the brain and the spinal column. They are definitely enriched there. They are enriched in lymph nodes. However, they can be detected in muscle, right? And so this manuscript, this paper describes how we developed a protocol to actually detect those prions. And this was led by Mancy Lee, who is a PhD student in MinPro Lab. She is supported by LCCMR funds, absolutely brilliant student who was trained in RT-QuIC at Colorado State University and has helped our lab to get that up and running. And so Mancy Lee is first author of that manuscript. But I believe we've also shared this with the committee for their reading. For developing new diagnostic tools. So we have said from the start that MinPro needs to develop revolutionary new diagnostic tools that are capable to detect prions in a variety of settings, including the field. And we believe that we're the first team in history, thanks to funding from LCCMR, the legislature, the Minnesota Agricultural Experiment Station. We were able to develop a nanotechnology-based test, a test that uses nanoparticles to detect prions in lymph node samples. We've been using this for lymph tissue and brain samples. We deployed this test in Rushford back in March, and we believe the first team in history to actually test for CWD at a field location within 24 hours. The output of the test is a color change. And so you can see at the bottom, the red samples there are, we have a positive control and a positive sample, and then the purple or blue are the negatives. We have a scientific publication that's ready to submit on this, and it must be further validated. This is not something that's going to be released immediately into the public. It's got to be further validated by other labs, but also we are working to identify industry partners that can really scale this up and make it more widely available in the future. But really, MinPro has a broadening research portfolio. So, I mean, what I've able to share with you all today is just a tip of the iceberg. We're partnering with prion experts. Dr. Jason Bartz is a close collaborator of ours at Creighton. Also, Dr. Joel Peterson, who's now at Johns Hopkins. We're working with scientists of the North America CW Research Consortium. Diversity, a large diversity of projects. We have at least 3 other new diagnostic techniques that have great promise. So we are engineering new antibodies, we're leveraging microfluidics, but also these nanoparticles that we have identified we're using in different ways. So we anticipate there's going to be more in the development of new diagnostic tests that will help revolutionize CWD diagnostics over time. And in this figure, I'm just highlighting some of the areas that I'm unable to go into a lot of detail on. We do have rapid PRNP genotyping. So this is something that's of great interest is, can you genotype the gene in white-tailed deer, let's say, that makes them more or less susceptible to CWD? We have rapid genotyping method that we're using with some success in the lab. but we're also pioneering forensics methods and environmental monitoring. And so there's some really exciting research avenues coming out of MinPro. All right, I'd like to shift gears a little bit and talk about some of the forensics research, if you will, of the Beltrami County deer carcass dump site. And we'll be hearing more about this, I believe, from the DNR later in this session. But just to recap, to make sure everyone's on the same page here, it was back in April that a 3-year-old doe tested positive from a Beltrami cervid farm. It was during that detection and the interviews that followed that the DNR and Board of Animal Health learned that there was a carcass dump site associated with the farm where the farmer was depositing carcasses within about a half mile, I believe, of the main farm itself. During that time frame in April, it was decided based on advice from the Board of Animal Health, the USDA, and the DNR that there needed to be a fence constructed to protect this site, this dump site from the resident wild herds. Then in May of 2021, early in May, our MinPro team, requested by the DNR, we were granted permission to access the site and collect material. We secured carcass remains, mainly bones, but also soil cores from the site, and plants that were growing or within or around carcasses that we were able to identify, to secure those into a biorepository for CWD testing. It was then during that time of visiting the site, we observed several bones distributed all across approximately 10 acres. And just so everyone is aware that when the carcass is dumped out, and this happens to be a forested area, the scavengers will come in and pick apart that carcass, if you will, and really just spread it across the landscape. So we were finding things, as I'm showing you with the picture down in the bottom left, you can see there's a spinal column there, but you would find a skull, a scapula spread across the landscape. So we did the best we could to collect those samples, and the DNR did as well. The samples were brought back to the veterinary diagnostic lab, and you can see in the pictures in the lower right here of our MinPro team working those samples. We were able to age class the samples, and there's a mixture of fawns, yearlings, and adults. We can't really determine confidently how many the exact number of carcasses that were deposited at the site, just because it was so worked over by scavengers and there's material that is likely missing. But in early May, we did identify at least 2 bones that were CWD positive using RT-Quick. Okay, so we're able to sample like bone marrow within the bones to use for RT-Quick testing. And then it was also in May that the herd was depopulated. So there's a 13 total CWD positive deer, including fawns on the farm. I wanted to highlight that, yes, fawns can have CWD. They can be exposed in utero. It's a misnomer to think that fawns can't get CWD. That's not accurate. So from June to present, the DNR has erected a fence to contain the site. It's about 12 acres that they've protected, and they'll be talking more about the fence itself. But our team secured additional funds to test all of the recovered remains and the soil from the dump site. So to date, we've identified at least 15 bone and insect samples that are CWD positive by RT-Quick. This is out of around 65 samples tested to date. What does that mean? Well, we've identified connective tissue, nervous tissue, bone marrow by cutting open the bones and sampling the bone marrow dust, if you will, that's left behind. We've actually taken skulls and performed a rinse in the skull using methods that were advised to us by Dr. Bartz at Creighton. And we're showing that, yes, we can use that to detect CWD prions in the skull. We did identify pools of maggots that were working on some of these remains and identify prions associated with those maggots. And we're working now in a sort of forensics entomology approach to identify the species of maggots so we can learn more about its natural history, what fly this might be. And so that's work that's going on in MinPro right now. But the material that we identified as positive was this cross-section of ages, included fawns, yearlings, and adults. What's very important is that we took soil cores underneath the remains that we were taking off the landscape. And so when we identified them as positive, we went back and found, took those soil cores out of our biorepository. We shared those with Dr. Joel Peterson, who is the world's authority on detecting prions in soil samples. He was at the University of Wisconsin at the time. His lab identified that the soil cores that we took beneath positive bones were in fact positive for CWB prions. So this means that the prions, as I've mentioned to the committee in the past, these prions can remain potentially infectious in the environment for years. And so we did identify those in the soil cores associated with the positive bones. What's concerning to us is that we also identified CWB prions in areas that we sampled that did not have bones associated with them. So we stepped some distance away. There were no bones on this particular area that we sampled. We included soil cores from that to the lab, and they identified them as some of those as positive for CWB prions. So this suggests some level of environmental contamination. Within the farm itself, there were 9 decomposed deer that were recovered during the depopulation event. Those were 2 decomposed for testing using traditional methods. So we tested those samples with RT-Quick and identified that 6 of those deer were positive. So that brings the total number of deer within the fence that were CWB positive to at least 19 animals. We say at least because there was other carcasses that we didn't have access to, but at least 19 animals we believe were positive in the farm itself. And I believe this is the last slide. So we are concerned about environmental contamination across the stump site. This is why it was so important that the DNR erected this fence. There is heavy usage across the site. So I'm showing you a satellite image of the site itself. Notice that there's agricultural land here. When we were sampling the site in June, there were deer bedding down. This is before the fence was erected. There were deer bedding down within this area. There are active, we were picking up fresh feces. And I believe the DNR folks on the ground noted that there were essentially waves of deer going back and forth in the evening to feed on the agricultural land and then right on the alfalfa field and then come back. So this is exactly the scenario that the DNR, USDA, and Board of Health are really worried about because of potential exposure. The areas in red are areas where we identified CWD positive material and or soil. And this area right in the middle is the primary spot where the dumping occurred. We believe this is where most of the remains are found. This area is contaminated with CWD prions. So we're working now to determine the extent of that contamination, both inside but outside of, and also outside of the fence. This, to our knowledge, this a study site like this has not been identified in a natural situation before. So it's very, very important that we maintain this fence, that the MinPro team gets long-term access to it, because we believe this is the start of a multi-year effort to better understand how CWD prions can last within the environment here in Minnesota, but whether or not they can remain infectious. And with that,
we're going to have to take the questions. I think.
Open to questions. Thank you.
Representative Eklund.
Thank you, Mr. Chair, and Dr. Larson. Thank you for the good work that your center is doing on advancing our knowledge of prions and their movement and what it means to our whitetail here, both farmed and wild. First question, Dr. Larson, is how much per animal does this RT for a live test? Does this RT Quick cost?
Chair Hansen, members of the committee, the test is about $14 a sample, I believe, and so that is so we've run the numbers. It's about $14 a sample. That number can go down as we ramp up, but that's about ballpark what it is.
Okay, Mr. Chair, thank you, and that certainly seems like a reasonable amount for a cost for something that's potentially so causing so much problem in our state. Dr. Larson, what kind of reception have you heard from the industry? Because all along since we've been doing this, all we've heard about is people saying we need a live test, we need a live test. Well, now we have one. So I'm wondering about the response you've gotten from the industry as to, are they going to get their are they willing to get their herds tested so we know exactly what's going on out there in the farm whitetail industry? Thank you, Mr.
Dr.
Chair.
Larson.
Chair Hansen, members of the committee, Rep. Eklund. Yes, we have shared this information with the deer farming community. I've talked of this live animal testing several times with the Board of Animal Health. And the Board of Animal Health, it can, in the language with respect to CWD diagnostic testing, the board can select a test and use outside of the need for USDA validation. So I believe the issue that we're working with is how there are concerns of privacy because it is such a new test and it's not been officially validated by the USDA. We can validate it internally. The question is, if we just start to detect positive animals in those farmed herds, what happens, right? What if it's not validated? What happens? Where does the information go? Is it reported to the board? And so there's concern there by the cervid farming industry about how to handle that information. And there are ways that we can handle that and secure, alleviate privacy concerns. There are similar tests that are done in swine and poultry for a live animal, if you will, tests for a variety of pathogens where privacy is maintained and where research teams like ours can work directly with the producers. So it is, I have shared this information with as many folks as I can, and we are happy to engage as time goes on.
Representative Eklund, final question. Follow up.
I'm good, Mr. Chair. I just, a quick statement. We've been hearing we need this live test. It's here. Let's see what we have going on in the industry.
Representative Fisher.
Thank you, Mr. Chair. Thank you, Dr. Larson, for being here today to update us to what's happening. My questions are along the line is, first of all, we're hearing about the live test. What is your capacity? How many tests are you able to do on a regular basis? And how quick is the turnaround?
Dr. Larson.
Chair Hansen, members of the committee, Rep. Fisher. Our MinPro is a research laboratory. The official diagnostic testing within the state of Minnesota is done at the Veterinary Diagnostic Lab. And so we have shared this information of RT Quick with the director, Dr. Jerry Torson of the lab. And they are, since it's so new, they're deciding what the demand is and how to onboard that, if you will. So in our research lab, we are able to do about, I believe we're up to 200 samples within 48 hours. We are securing additional machines to ramp up our RT Quick throughput. But again, we're a research lab. If the state invests in RT Quick technology with the Veterinary Diagnostic Lab, then that would provide really the infrastructure. And you really need to have the official infrastructure that the VDL provides to be able to do this adequately. But there's a transition time where MinPro could help, right, train and get the VDL up and running with that.
One follow-up, Representative Fisher.
Certainly. The other follow-up I had is, besides the live tests you're mentioning, you have tests where you can go in and sample the soils, et cetera. Has there been any discussion to take a look at going through and being able to test not just the live animals, but what might be in the soils of all the farms that are currently out there? Because we know that an animal gets sick, it may be a couple of years before they start showing symptoms, and it could already be in the soil. And so is there anything being looked at to try to address the soil and the plants and within the farm enclosures themselves to see if it's already embedded within the internal structure, the biological structure of the plant life or the soils, to see if we've got pending issues down the road, even though the animals themselves may not be yet showing it.
Dr. Larson.
Chair Hansen, Rep. Fisher, members of the committee. Yes, that's a possibility. The issue is that we would need to have permission, right? This is private property. We would have permission to access those areas. I strongly believe that we could have prevented what happened in Beltrami County with a pre-movement live animal test. But there are environmental options with this. And the issue is working with the cervid farming community to identify the best way forward But you're absolutely correct that we could sample all farms right now and potentially detect the CWD prions using this approach. And so that's something that we just have to, I think, proceed with caution and think through. But we can see that there's other states that are with a growing interest in RT Quick. Texas is one, for example. And so I believe we're in this transition zone where this new technology is providing new opportunities, and we have to understand how we can best use it to protect all cervids.
Representative Becker-Finn, and then Representative Lippert, and then Representative Heinzman will have the last question for Dr. Larson. Representative Becker-Finn.
Thank you, Mr. Chair. And I want to go back to something that you stated earlier, Dr. Larson, about being able to find the evidence of CWD prions in muscle. And I just want to, for folks' benefit, if you don't think too hard about your meat, that's the part of the animal that you eat. So I wanted to highlight that, and just if you could just clarify that, but then also what are your recommendations then for hunters and those of us who butcher our own animals as far as what we should be doing to keep ourselves safe as we hunt and got the animals in the field and then prepare that food for our families.
Dr. Larson.
Chair Hansen, Rep. Becker-Finn, members of the committee. First, it is critically important to keep hunting. We have to keep hunting in order. It is a tool to fight this disease. If hunting erodes, then there's data out there that shows that CWD can expand. Yes, we can use this technology to identify prions in muscle, potentially venison. What we're thinking is that can eventually be used. It's going to take time. It's not anytime soon, right? This takes years to develop and unroll, but that could be used ultimately as a food safety test to help hunters be more confident in, or any venison consumers more confident in what they are consuming. What we recommend is to follow the latest advice from the DNR. What's on the DNR's website? Know where CWD is in the wild. The DNR has done a fantastic job of surveying wild populations, right? And so hunters can use that information to look at and see where CWD is in the state. And they should follow the DNR's recommendation for testing their harvest. When you do those things, the hunters can be more confident in what they are consuming.
Representative Becker-Finn. Quick follow-up or?
Yep. Thank you, Mr. Chair. So as far as practically, what does that look like for us? Is that wearing gloves? If you think through someone butchering their deer in their garage, what do we need to do so that we are being as efficient and safe as possible when we're dealing with those animals?
Chair Hansen. Members of the committee, Rep. Becker-Finn. Yes, we recommend wearing gloves, right? Like latex gloves or nitrile gloves as a PPE. And I believe that the DNR recommends that no matter what, when you're processing your animals as the first layer of protection. If you are harvesting animals that are within an area that the DNR has shown has CWD at some level circulating within the wild, we recommend using a 40 to 50% bleach solution to clean stainless steel. So knives and whatnot can be cleaned using a 40 to 50% bleach solution. That does not work that well, though, on porous materials. So plastics, wood cutting surfaces. That's more difficult to treat for prions. But stainless steel, a 40 to 50% solution, and then wear gloves.
I'm going to move to Representative Lippert, Representative Becker-Finn, or you can state your question and we can get a response in writing for it back.
Yep. Just last question. This might be really quick. Is what do you think the chances are that wild deer in that area near that Beltrami County dump site have gotten CWD?
Dr. Larson, do you want to take that one now? Or?
Chair Hansen, members of the committee, I'm concerned based on the potential environmental contamination that we've identified and also the amount of deer that were on that landscape. I hope that we caught it in time. We will see in the next 2 or 3 years whether or not it's circulating in the wild based on the DNR surveillance effort.
Representative Lippert.
Thank you, Mr. Chair. Thank you, Dr. Larson. I was concerned when you mentioned the positive insect samples, and I'm wondering if you could just talk more about the significance of the positive samples and what that might mean, if the maggots or flies might transmit CWD. Just wondering if you could talk through the significance of that.
Dr. Larson.
Chair Hansen, members of the committee, Rep. Lippert. We were surprised to get that result, but we wanted to. We know that other scavengers, there's in the literature, it's showing that coyotes, for example, you can detect the prions in the feces of a coyote that is consumed in a positive. Also, that the prions can be distributed around the landscape by rodents. To our knowledge, we're the first team that has identified these prions associated with an insect. As you can imagine, the small body size of an insect, we're assuming that the maggots are a species of fly. The number of prions on a fly is going to be very small. So we're thinking of this more of a surveillance, where we can go in and potentially collect insects on this landscape, and we can identify whether or not they are being exposed to prions just by crawling around on the soil or the plants and whatnot. So this is very much in its infancy, but I do not think that based on what we're seeing thus far, we have to generate more data, that individual flies are of concern for transmitting this. It's more of a, can we detect the prions on them to see if it's within the landscape?
Representative Lippert.
Thank you, Mr. Chair. Thank you, Dr. Larson.
Representative Heinzman.
Thank you, Mr. Chair. So Dr. Larson, I had a couple questions. Number one, have you started testing outside of fences, looking in the wild? Because that's, of course, a very likely possibility that if we have prions in soil that you've confirmed with the RT quick test, I'm assuming you're checking outside of the fence to look to see that it is potentially in the wild. We talked about that at the LCCMR when you presented years ago. And the idea was you could be checking feces for the presence of CWD out on the land. And of course, that's a whole nother conversation that this committee will likely be having for years to come. So that question, and then I'd also like to know, what is the potential percentage, or what is the percentage of false positives that RT Quick has generated? And I know that's a question of accuracy, but I'm sure that's something that's happening in the lab. You're confirming what is the potential accuracy of RT Quick, and that, of course, would call into question some of its ability to confirm this. So those two questions are real important to me at the moment. So if you could touch on those, that'd be great.
Dr. Larson.
Chair Hansen, members of the committee, with respect to your first question, Rep. Huntsman, so to be able to test test feces, outside of the fence and in the wild, remember that in the wild herd down in the Preston area in the southeastern part of the state, it's about a 1% to 2% prevalence rate, and so to be able to effectively go out and secure fecal material from that population, we would be generating RT results on many samples with only a relatively few positives. And so the best way to proceed with fecal testing as part of the live animal approach would be to actually be able to test samples within the farmed herds, right? To be able to understand that's a smaller number of individuals that we'll be dealing, and then we could look for potential CWD circulating within herds within a given farm. Now, it is possible to take environmental samples on public lands or private lands that we have access to downstream of cervid farms that have gone positive in the past, so we could better understand whether or not those prions are circulating in the wild, if you will. And we do have funds from the state to look at water, for example, to see if we can detect the prion circulating in water downstream of point sources of infectious prions, including in the wild herds, right? So we'll be doing work in Winona County to look at, can we detect CW prions downstream of areas that the DNR has identified CWD to be circulating with greatest frequency in the wild herd. The second question with the RT Quick sensitivity specificity, we've shown using a sample of about 500 deer through collaboration with the DNR, that RT-QuIC was 100% accurate in identifying the animals that had CWD when using the same tissues that the DNR used, so retropharyngeal lymph nodes, for the traditional tests. So 100% accurate with respect to the same tissues in comparison to the traditional tests. False positives. So the issue of false positives really depends on the sample type that you're testing. There could be more false positives, for example, using plants or environmental samples, but that rate is still going to be very low, maybe less than 3%. But there are methods that we can use to reduce the chances of false positives. And we also have control samples in every test that we perform. So thus far, we have had almost no indication of false positives. If anything, there can be a false negative rate with RT-QuIC, depending on the sample type. But the test is just, it's a very robust assay all around.
Mr. Chairman, a very quick follow-up. It's a yes or no question.
Yep.
So on my first question, the question was very specifically, are we or have we yet tested outside of a fence? So in the wild, I think that's a yes or no question.
Chair Hansen, Representative, yes, we have used RT-QuIC to test wild populations. Yes.
Representative Heitzman.
So that maybe I wasn't specific, and I might just be hearing you incorrect or wrong, Dr. Larson. In the wild, I mean, so you're checking deer specifically, or some of the other samples that you were talking about, soil, insects, feces?
Chair Hansen, members of the committee, we have tested feces, fecal samples from wild deer, blood samples from wild deer. Thus far, there's been no reason to test insect samples from the wild, just because we're dealing with a very special case here in Beltrami County, where there's the concentration of carcasses on this one spot of land. But yes, we have tested fecal samples from the wild. With respect to soil, again, it's a matter of, is there a particular area of intense interest, maybe that the DNR has identified with a lot of positive deer associated with it in the wild? We could take soil samples from there, but it becomes more of a fishing expedition, if you will, just because of the wide variety. Like, how do you actually pinpoint and do that? And so, yep.
Mr. Chairman, just a quick comment. Thank you for the opportunity to hear this testimony today. I think obviously everybody is wanting to know where the CWD may potentially be, not only on farms, but in the wild. That's a question that's come up frequently in greater Minnesota. And the containment fence in Beltrami is obviously an attempt to limit access from wild deer to coming into that area and potentially being impacted. So it's super helpful to get that feedback, Dr. Larson. Thank you for that. And thank you, Chair Hansen. And hopefully the testing isn't considered a fishing expedition, but rather just a scientific fact-finding mission to determine where CWD is. And if it is out in the wild, in the landscape, in the deer swamps that we're all hunting, we'd like to know. So hopefully those tests are forthcoming. Thank you.
And members, I was giving a little more time here because I think it's really important. We are running behind about 15 minutes. Next up is the Minnesota Department of Natural Resources and Dave Ofelt. If Mr. Ofelt could introduce himself, and each of the team members there could introduce themselves as they speak, and we will begin. We're targeted to be done about 11:05 on this.
Thank you, Chair Hansen. My name is Dave Ofelt, and I serve as the Director of the Division of Fish and Wildlife at the Minnesota Department of Natural Resources. We're pleased to be here today to give you some updates on some agency work around CWD management. And we've got a lot to share and not a lot of time to do that, so I'll be brief. Before I make some introductions, I want to thank you for the support, the ongoing support for this really important work that is going on in the agency, and appreciate the chance to be here. Primary presenter today will be Dr. Michelle Carstensen, who is the supervisor of our wildlife health unit. She'll be giving most of the update. She'll be joined partway through by David Benke. Mr. David Benke works for the Pollution Control Agency. They'll be talking about work around carcass disposition. And then a little bit later, Michelle will be joined by Mr. Chris Balzer, who is a DNR area wildlife manager in our Cloquet area and one of our staff people who are taking the lead on working with our colleagues on the Board of Animal Health on concurrent management. So with that, I will pass the baton to Dr. Carstensen. Thank you.
Welcome, Dr. Carstensen. If you could introduce yourself for the record. And I think one of the important things, the archery season opens this weekend. Is the DNR and the PCA ready for that? So, Dr. Carstensen.
Thank you, Mr. Chair, members. Thank you for having us here today. My name is Michelle Carstensen. I'm the Wildlife Health Program Group Leader, and be kicking off today's discussion on what's happening on the wildlife side with chronic wasting disease. The cover slide that I have here that you're looking at right now, hopefully that's coming through okay, depicts all the deer in this photos are all CWD positive Minnesota deer. So you can see a diversity of what they're looking like to the hunters or the members of the public that found these animals and reported them to us. So just to show you that variety. Briefly, just to reorient everyone about our current status about this disease in Minnesota, I have a slide here that's updated to show counties in green where positive cervid farms have been discovered since 2002, and there's been a total of 12. And also the red dots depict areas in the wild where wild positives were discovered. Again, since 2010, which is our first one in Olmsted County. And then the majority of positives that we've found have been 2016 forward in several additional counties, primarily the Southeast and South Metro. Just a quick look back to last year, just to remind folks that we did do a full voluntary sampling last year due to the COVID-19 pandemic. We switched gears from what we had planned with some opening weekend mandatory collections to all voluntary with self-service stations to minimize contact with hunters from each other, as well as with our staff and students that do this work. We did collect over 8,000 samples. We detected 27 positives. All of these positives were in the Southeast and South Metro management zones. We did do a sufficient job in collecting information from our management zones, but we did fall short in some of the new surveillance areas, particularly the Pine County and the Douglas County areas that were new disease detections and captive service facilities. We didn't collect enough information there to really inform what's happening in the wild last year, but we will hopefully regain that confidence this coming fall. So just a quick slide to show again where we have this positives in the landscape for wild deer. As I mentioned, this past fall, we had 27 new detections. The majority of those, again, are in the Southeast, with 3 new positives in the South Metro, which is our newest area in Dakota County. We have a total of 5 cases there now. Some good news is the North Central zone, which is the area around Brainerd. We still have not detected disease except for that one positive that was detected just less than a half a mile from a positive cervid farm that was depopulated in 2019. So if things go consistently well there, this would be potentially the last year of conducting surveillance in the North Central area. And just to make sure folks realize too, the level of prevalence we're talking about, there was a lot of dots in the Southeast that look a little overwhelming in the last slide, but just to scale that up to what we're talking about is really a low prevalence of disease on the landscape in the wild. As Dr. Larson mentioned, less than 1% is our average point estimate in the last 5 years. And this is in our Fillmore County outbreak where we're finding the most disease. So we're testing thousands of animals in this area and finding low prevalence. So it appears to be persisting there, but this is also still a rare disease in Minnesota, and we're trying to keep it rare if we can't actually go backwards and extinguish it altogether. So looking forward to this fall, we have 6 areas of the state we intend to be conducting surveillance. The newest one, as we've been discussing, is the Beltrami County area. We will be conducting mandatory sampling over opening weekend of firearms A and B seasons, with the one exception of the West Central area, which we're implementing our 2nd year of a risk-based pilot under a different point scheme scenario to collect information. That will be also happening throughout the season for that area. In our CWD surveillance and management zones, management and control zones, there will be self-service stations available throughout the hunting season, as well as dumpsters to accommodate carcass remains. In our surveillance zones where we don't have disease yet confirmed in the wild, that again is focusing on opening weekend mandatory sampling. And if hunters would like to contribute samples outside of that time frame, they can do so by appointments with our wildlife offices and still have those deer tested. We're expecting over 22,000 samples this fall and a cost at roughly $2 million to do that work. And implementing that takes a lot of coordination and a lot of sites that we need to be present at and work so that we can have the best access to our hunters. So that's going to include 87 total stations in our areas that we're going to do sampling, and another 14 in the West Central under that risk-based model. And given what we learned last year in our all voluntary efforts, that wasn't sufficient to reach our targets, but it also did allow hunters to sort of choose how they want to interact. If they want to continue with self-service due to risks associated with the ongoing pandemic, they can do so. We have about 30% of our sites that are going to be self-service only, and the remaining 70% will be staffed, where there can be in-person interaction with DNR and student labor. Again, we have over 400 folks that will be working to make sure this effort is successful and that we can be there to collect the samples that we need. Just a brief overview of regulatory changes that go along with CWD management and surveillance. This slide's a little bit busy, so I just thought I would hit some of the highlights. Again, we have that mandatory CWD testing on opening firearms. We also have a reduction in bag limits in the management zones from unlimited to 5 deer, which isn't really expected to influence anything about harvest. It's more of a public perception that DNR is trying to eliminate deer populations. That's not what we're trying to do, just trying to give a lot of opportunity. So that change won't really affect any hunters, as 99% of our hunters are really within that 5 limit bag already. And it does not apply to the early antler lists and late season hunts. There's information about the late hunts in the regulations booklet this year, so hunters can already be planning ahead for participating in those late season hunts. And we will be expanding our deer feeding ban with the new detection of disease in Beltrami County. And work is going on right now with our commissioner's office to determine exactly what that footprint will look like. We also have the Adopted Dumpster Program back this year. This again began in fall 2009. This is available in all of the areas where we have carcass movement restrictions. So that's our CWD management zones and control areas. We have 32 dumpster sites that are being planned. Bids are out for most of these and received. We're still waiting on some bids for the new Dakota County area, but we expect to have everything ready to go for this weekend, opening archery hunting. And we then expand to additional sites during the firearm season as well to make sure that hunters have easy access. And this has been a very popular program. Even last year when we had lower participation rates in sampling, we still had a lot of usage of the dumpsters. Over 200 tons of deer waste were hauled to landfills with this, this program. And just lastly for me on this topic about carcass disposal is really just kind of telling you what we want of the hunters in this framework is we really want them to do 3 things: register their deer after harvest, and they can do this in three ways: with phone, online, or walk-in big game stations. We want them to comply with the mandatory testing, and they can choose either a self-service option or in-person sampling at check stations. And we want them to comply with the carcass movement restrictions in our CW zones to not take those intact carcasses out until they receive their test results, which again is 4 to 5 business days, which really translates into a week given the weekends that are occurring during deer season. And it can be up to 2 weeks if there's a really busy peak at the lab and they get backed up. So just to make a plan, and they can do that in these zones. They can use meat processors. We have them identified in each of our surveillance zones, our management zones where these restrictions are in place. There are options for meat processors. You can still hang deer at camp, like maybe hunters have been doing for generations, and await those test results, and then natural decomposition is okay. And also, we can use the quartering setups at our dump stations and quarter the deer right away, and that meat can leave the zone immediately, and the remains can go in the adopted dumpster program that we have set up. I will switch quickly to Dave Benke.
Mr. Benke, if you can introduce yourself for the record.
Thank you, Mr. Chair, members of the committee. My name is Dave Benke. I'm the Director of the Resource Management and Assistance Division at the Minnesota Pollution Control Agency. So once we get the animals harvested, the question remains, what do we want to do with the carcass the butcher remains, and so forth. When we look at this, we look at it in a couple different ways. We want to look at the surveillance and the management areas just a little bit differently. But what we want to make sure is that there's an appropriate management method available for the hunters to dispose of their deer carcasses. Keeping those carcasses off the landscape is really important, and providing those opportunities for disposal is key. The testing that Michelle talked about can determine the highest risk, those infected animals, but sometimes that testing is a little bit behind in terms of timing, especially during the heavy firearms season where most animals are harvested. So we know that some of the material may end up in the solid waste stream, end up in dumpsters. We want to make sure that the disposal methods that they use for regular waste are still available throughout the season. If there is an infected deer, and Michelle's alerted to that, I'm sure she's able to make arrangements to remove that. But like I said, not all of the animals necessarily will be under that circumstance. The next slide, please. So in the surveillance zones, East Central, Pope Douglas, and the new Beltrami, Polk County area, we've been working with the counties on each one of those facilities to make sure that they're available. Keeping the proper disposal options available in the current waste system is what we're after, so that the public, the butchers, the meat processors, the taxidermists all have that system available. We've met several times with the East Central Solid Waste Board to make sure that as long as the disease has not been confirmed in the wild, those facilities are still under use. If it does get confirmed in the wild, we may have to go to a different understanding and a different option. But for now, these three areas still have the disposal facilities available for this level of surveillance. In the Pope Douglas area, we're using the waste energy facility to manage the butcher remains, the carcasses. And in the Beltrami, Polk County area, it's a joint use of the system. So the Polk County landfill has been the designated location where the materials will end up. Polk County does run a waste energy facility, but during the height of the hunting season, they have already provided dumpsters for disposal of carcass remains, not just from deer, but from moose, from bear, and from other wildlife that are harvested. So they've been doing that, and keeping that facility available is the goal there. In the management zones where we have disease confirmed in the wild deer, those areas with carcass movement restrictions where everything has to stay in the same area, the disposal locations that are needed have kind of been looked at as a little bit more long-term. The public, the butchers, again, the taxidermists, and now the dumpsters from the collection sites are all places where we want the waste to be deposited from those folks. And so we want to make sure that those are available. In the Southeast, we worked with the Olmsted solid waste folks and the Olmsted County Board to get the waste energy facility available and also maintain the use of the La Crosse landfill as we have in the past. So now we've got a couple options that are going to help us manage the deer remains in that area. In the Crow Wing area, as Michelle mentioned, the Crow Wing County landfill has an air curtain incinerator there, and this may be our last year of using that unless the county decides to continue to run that program. So the deer in that management zone, butcher remains, all end up at that facility. The air curtain incinerator incinerates those, and then the ash is deposited in the Crow Wing County landfill. In the South Metro, in that management zone, we use the Republic landfill as part of the disposal system there for those remains. So we're ready to go. We've got places for the material to go, and we'll continue to have the talks and discussions with the surveillance zone impacted systems and make sure that we've got a place for the materials to go that reduces the risk to the lowest possible. So I'll turn it back to you, Michelle.
Okay, thanks, Dave. And thanks, committee members. Just a quick recap on spending. Just wanted to show here the highlights of the last almost 2 decades now of CWD work that we've been doing. Has a total price tag of over 14 million. This past fiscal year, we spent just over 3 million. And there's a breakdown here to kind of highlight where those resources were used between our sampling efforts and salary and diagnostics and dumpster program, which is about 230,000 this past year. So just wanted to let folks see what the spending plan has been like. And then to kind of circle back to the fence that Dr. Larson talked about, again, we responded to that information about carcasses being disposed of on county administered land and quickly worked to determine the level and extent of contamination and partnering with MinPro to collect those samples, as Dr. Larson already talked about. So this is sort of the footprint here of what the fence ended up looking like. So it's about 12 acres to contain the highest risk areas. That includes all of the sites that Dr. Larson had identified as also suspect for prion contamination in the bone samples that he was mentioning. And just a couple photos here to close out my portion here to just show what that fence looks like now, completely constructed. We did so as quickly as possible. The weather actually played to our advantage because this is a wet, can be a wet area within this complex. So we weren't sure we could actually go in and build a fence this time of year, but the drought helped us get ourselves in there to do this work. And cost us approximately $194,000 to construct this fence. About half of that was DNR salary because we built it ourselves with our own labor force, with the one exception of a contracted vendor to pound the 16-foot posts in. This is a 10-foot fence meant to last 20 years because we wanted it to be sufficient to contain risk well beyond the current period of 5 years that USDA recommends, and also beyond the 16-year window of scrapie in sheep where it has remained infectious. So we wanted to do our due diligence and contain this the best that we could. With that, I will switch over to Chris Balzer to talk about the co-management.
Thanks, Michelle, Mr. Chair and members of the committee. Yeah, my name is Chris Balzer. I'm the area wildlife manager up in Cloquet, but currently working as a project manager on co-management of captive whitetail deer farms. So why are we here? You know, I think folks are aware of the recent legislation that was passed that gave co-management authority to both the DNR and the Board of Animal Health, and that's for farmed whitetail deer. Part of that legislation provides for or asks that the Commissioner of Natural Resources, in conjunction with the Board of Animal Health, must submit a report to the chairs and ranking minority members of the legislative committees and divisions with jurisdiction over the environment and natural resources and agriculture on the implementation of concurrent authority under this section. So that report must include a summary of how the agencies work together under this section, including identifying any challenges, an assessment of ongoing challenges to manage chronic wasting disease in the state, and recommendations for statutory and programmatic changes to help the state better manage the disease. Do I have the ability to switch the slide? No. No. Okay, here we are. We're kind of still figuring this out. I mean, co-management is a new thing, so we're getting our feet on the ground, but our progress to date, the big thing was developing a project plan, which is identified. This is our DNR plan on how we want to attack this or proceed with it. It defines our goals, the deliverables, timelines, and staff roles. As part of that effort and to inform the producers out there, a joint letter from the Board of Animal Health and DNR was sent to all whitetail deer farm producers, kind of just letting them know, make sure they're aware that it's now co-management and what they might expect. And currently we're going out on whitetail deer farm inspections with the Board of Animal Health. We've had several completed already and several scheduled. We set an internal goal as part of that project plan to do 40 inspections prior to December 17th. December 17th gives us some time to kind of analyze that information and provide input into the February 1st report. And we're also working cooperatively with the Board of Animal Health in white-tailed deer escapes and follow-ups on any deficiencies and enforcement action that may be required as a result of those escapes. So that's all I had. Any questions?
So we have 3 questions so far, and members, we have a little bit of extra time, so we don't have the hard stop at 11:30, but I want to make sure we get the public testimony in. So Representative Becker-Finn.
Yeah, thanks, Mr. Chair. And first, I want to point out, I heard multiple times reference to the surveillance zones where we don't have CWD in the wild, and that isn't accurate. We do have CWD in the wild. We just heard from Dr. Larson that they tested multiple places where there was CWD in the wild. I understand that we haven't confirmed that yet in live animals, but there certainly is a much bigger risk than a typical scenario where it's within a farm, and we've treated that, the lowest level of surveillance. And what that means in practicality, and I want to walk through this as a deer hunter. So my kids eat venison at least twice a week. I've said this multiple times, and it's true for many families in rural Minnesota, that venison is the primary protein source for my family. And so now we have this dump site and this not good situation that's close to places where a lot of people hunt. And so because we've decided to categorize this as a lowest surveillance zone, if I get a deer outside a Saturday and Sunday that first weekend, I'm now looking at calling to make an appointment and then driving a sample to Bemidji or Park Rapids, and then hanging my deer separately. But if it's as warm as it was last year, it's not going to keep. So I'm going to wait for 2 weeks to get the results of the CWD test, maybe, but I can't let the deer rot or that meat isn't edible. So then my option is to figure out a way to refrigerate it. But I also just heard from Dr. Larson that plastics and other things aren't good and we should be using stainless steel, and then we're going to have to disinfect with 40 to 50% bleach. This isn't going to work. And I think that we need to try harder. I think we've got to figure out a way, whether it's refrigerated trucks or getting a contract somewhere, whatever we need to do. But for folks who really care about this and want to keep their families protected from eating the muscle where CWD prions can be, it isn't reasonable to ask a person to butcher something at home with their bare hands and then wait around for 2 weeks to maybe be told to throw it out. And now I'm being told I should be driving it to Foston, which is 90 minutes away from where I hunt, to go dump all the meat I just carefully processed and wrapped in paper and labeled for my family to eat. So I just need people to think that through, because I don't think that we are. And think about it not just from a disease, like scientifically this is interesting, but I'm a family trying to feed my kids. And I don't think we're trying hard enough to figure out a better solution for folks who want to know whether that deer has CWD. And I think especially because we've done no testing in Beltrami County, and this is a new scenario where we've got public land where it was dumped, where we know CWD is, where we know deer are, we know how far deer travel. I'm just very concerned that nobody is thinking this through to the degree that many people like me are thinking it through. And my family in rural Minnesota, and particularly our families who rely on this for food. So not even getting into the cost of what that's all going to be in my time. And I can't go buy, I don't have $1,000 to buy 80 pounds of organic venison from a farm. So just, I'm wondering if you had the funds, which I think you should, based on all the money that we've been giving to the DNR for this, what else could we do to make this easier for people to get their deer tested who are within proximity to that Beltrami site? I'm asking you right now to try to problem solve this. What are some ideas that we have for how we can do better for deer hunters and families who rely on venison for food?
Dr. Karstensen.
Mr. Chair, members, Representative Becker-Finn, thank you for your question. I think I should maybe clarify that if you are a hunter and you have a concern about that venison, your deer being tested in any of our surveillance zones, you can actually make an appointment at any wildlife office, not just ones in the zone. So depending on where you are in the metro or at camp, you might have a closer option to travel, but that isn't 90 miles. But just getting back to our planning, we do have a CWD response plan that we're following with this situation as we have in the past. And we need to do the work to decide if we have disease in the wild. And I think we have had several high-risk scenarios since I've been here for 17 years doing this, including Pine Island, where we had a facility with 25 breaches in the fence, wild deer inside and out in a large facility. And I swore up and down we were going to have more disease in the wild and had a larger situation than we did. And it turned out we had one deer over years of testing. That's what we found. And so I don't think there's 100% certainty that things are that bad in Beltrami, but the risks are high. And I think the risks have been high in several of our recent situations where the disease has been found in the landscape. I think we need to do our work. And then if we find disease, we will bring the additional resources that go along with management zones to this area. And that's going to include carcass management restrictions, multiple bucks. And so when we bring all of those tools to the table, it has a lot of impacts to the hunters in this area that also are going to bring a lot of concerns. And I think we could alienate hunters by trying to do some of those things ahead of the work to show where the disease is or isn't, and also the scope of that. Do we have it at 1 deer, 15 deer, how large of an area? So I think we need to do our work to have the best informed decisions about the size and scope of that management zone. That will help bring our hunters with us and get behind us in what will come next, which is a lot more regulations and control of the hunting season and carcass movements and dumpsters and the like. So it's not just about the funding, as you pointed out, Representative. It's also about the commitments from our hunters and be transparent in why we're adding in these different layers into the regulations of their hunt that they're going to be seen in the future, and to not get ahead of that.
Quick follow-up, Representative Becker-Finn. We've got 3 other hands up. So.
Yep. Thank you, Mr. Chair. And, I am trying to partner with you guys, but that was not a response to my question. That was doubling down on the plan that's currently in place. And I am a deer hunter, and I am telling you, we need a better plan. It can be voluntary, but this isn't enough. Telling people that they're going to butcher an animal in their garage with their bare hands, and then they might have to throw it out. We're going to lose hunters if people don't feel safe that they can eat the meat. We need that for people. So the response plan was approved in July of 2019. It did not contemplate what happens when a bad actor dumps CWD carcasses on public land. That was not part of that wasn't in the scope of what we were doing. And so I hope that this conversation can continue and that you hear me. Your plan is not good enough. I am a deer hunter from rural Minnesota, and this isn't enough. I'm not saying we need to bring the hammer down. I'm saying we need to make options for the people who want to know so that they can feel safe and feel good about deer hunting and feeding their families. Why would I spend a whole week of my life out in the woods, working my butt off, doing all the work that goes into the hunting, and then not feel confident that I can feed that to my kids? We got to do better. And this is me asking you to be open to doing more than what is currently in your plan, because it's not enough. Thank you, Mr. Chair.
Representative Wozlawik, then Representative Jordan, and then Representative Acom.
Thank you, Mr. Chair. I have a couple of questions related to some of the sort of restrictions that are in these different areas when there is CWD detected or not detected in the wild herd. My first question is sort of how are these things enforced? So I'm thinking specifically of things that like the restrictions on carcass movement and the deer feeding ban, those 2 things specifically, because those are things that I think people can easily conceptualize. So if there can be how are we enforcing that to make sure that those things are happening in these areas where we have concerns about CWD. And then, so that's the first question, if someone can answer that, and then I do have one follow-up.
Dr. Karstensen.
Sure. Thank you, Mr. Chair, representatives. We don't have anyone from our enforcement group here today to talk about numbers of citations, but we do put carcass tags on every deer that comes through our stations, as well as at the self-service check stations. So they have an identifier that equates to them submitting a sample. And that's a really easy visual cue for conservation officers to inspect animals that they encounter to make sure that that testing was done. Hunters and enforcement alike can actually look up those test results in real time too. They have access to the online system. So if there is concerns about somebody removing an animal out of the zone prior to test results or that didn't comply with testing, they should have that ability to check on that with either the tag on the back leg or looking it up in the system. I think with the feeding ban and along with baiting activity, enforcement does regular flights to look for any of that kind of activity. There has been, baiting complaints every year. That's been one of the biggest issues enforcement deals with statewide. So I think they're using their resources the best they can to enforce that, as well as the report your neighbor kind of thing that happens when folks feel that neighbors might be using bait or attractants illegally to bring deer onto their property. That's often brought through the tip line. So I think enforcement is doing the best they can to try to enforce those regulations, and we will continue to do so.
Representative Wozniak.
Thank you, Mr. Chair. Just a quick sort of a follow-up or maybe an idea, but I'm thinking about prevention, right? So wanting to prevent, jumping off of what Representative Becker-Finn said, wanting to prevent the spread of CWD as opposed to just responding when there's an incident and then setting up a zone and that sort of thing when we confirm test results. Has there been any discussion on sort of expanding on these things that are currently in place, like a statewide feeding ban or something? I understand why we're focusing on these zones because we have CWD there, but it seems like if the goal is actually prevention, which oftentimes is much less expensive and just better in general for our natural resources and our wild deer herd, sort of what are the conversations around what are we doing to prevent? Are there conversations around making these restrictions apply to a broader part of the state, or what sort of things are happening on the prevention side so we can prevent the spread as opposed to responding to incidents?
Dr. Karstensen.
Mr. Chair, representatives, I'm actually going to see if Assistant Commissioner Bob Meyer might be able to help me out on this one to talk from the Commissioner's office standpoint about where we're at with the feeding ban, and I think interest in broadening that beyond what we have currently, as well as to a statewide scope at some point.
Commissioner Meyer, welcome and introduce yourself for the record.
Mr. Chairman, members, for the record, Bob Meyer, Assistant Commissioner, Department of Natural Resources, and thank you for that question. It is something that we do talk about all the time and trying to figure out how do you do that on a statewide basis. There's a number of competing interests, such as people who feed birds in their backyards. You have other hunting seasons that would be impacted, possibly other locations. Actually, I've had conversations with Representative Eklund about this as it pertains to facilities in his district. It's something we're interested in trying to do, but we've tried it before and we were unsuccessful in the legislature. So looking at how do we proceed, if we did it through rule, it would take a couple of year process to do that. But it is a conversation we're open to having with you all and talking about that. The other piece of that is scents and attractants as well, deer urine. We do prohibit those things in our zones currently, in some zones, by emergency rule that we set up by season. But it's a conversation, if we're going to go down that road, we need to have with the manufacturer of these scents and foods and attractants as well, and give people time to comply. So, but it is really important and something we are interested in working with you all on. So,
Representative Jordan.
Thank you, Mr. Chair. And I'm hoping we can maybe just take a little bit of a step back as we're looking into co-management. I have some questions just generally about how cervid farms are managed. So I don't know, now that we have joint management with the DNR and Board of Animal Health, who would like to go about doing this. So what permits are required by the state of Minnesota and how much do they cost if someone wanted to start a farmed deer operation here in Minnesota?
Mr. Chairman, Representative Jordan, I think that question will be best answered by the Board of Animal Health, which is coming up next to talk about those types of things. So,
okay, I have a question then just for the DNR. And I heard Dr. Carstensen give an exact number for the fence in Beltrami County. Can you repeat that number for how much the fence cost and how much the DNR spent on that? And then for everyone watching at home, I want to be clear that when we say the DNR is spending this money, this is money that's done by Minnesota taxpayers. So this is money that every Minnesotan is spending. So how much was the fence for the Beltrami County? And how much did the DNR pay for it? And how much did the farmer pay for this fence?
Mr. Chairman,
Commissioner Meyer.
That really quickly. The fence is approximately $194,000, which the DNR has funded to date. You'll see in the slide, it was a lot of labor and materials basically made up the two costs of that. And we are in the process of working with the board to recover those costs. The reason why the DNR paid those costs or spent those dollars now was to get the fence up as fast as possible. So we basically fronted the money to get the fence up, and now we're in the process of trying to figure out the best methods to recover those dollars.
Okay, so just to be clear, it was $193,000 from the DNR, and the landowner has paid none of that so far.
Yes.
Okay, thank you, Mr. Chair, and thank you, Commissioner Meyer.
Representative Acom.
Thank you, Mr. Chair. And I think actually my questions are going to be better pointed toward the Board of Animal Health. They're more about the licenses as well. So I'll maybe hold my questions.
Couple of quick questions. The authority that is joint there, so then the enforcement authority that is provided in statute to the Minnesota Department of Natural Resources can be applied to inspections and violations of captive cervid facilities. Is that correct?
Mr. Chairman, members, Bob Myers, Commissioner, correct that the authority actually goes to everything that the board has on white-tailed deer in those several sections of Chapter 35, including rulemaking authority dealing with CWD and protecting the deer. So we are working very closely, as Chris talked about, in establishing this and learning to walk before we run. And I think the cooperation has been great. We're getting access and to the database, the data we need. And we look forward to coming back with our report, which will really guide, in my opinion, the next steps of CWD management.
And Commissioner Meyer, have any of the captive cervid facilities refused the DNR access?
Mr. Chairman, we have clarified that the DNR does have that authority and sent a joint letter out. Some producers were hesitant, but now that they've received this communication, which we can forward to the committee, I apologize for not doing so, clarifying that we do have dual authority. We have not had any instances to date.
And have you received all the information you need from the Board of Animal Health with prior inspections? Is the data flowing back and forth between the two?
Mr. Chairman, yes, it is going very well. We're in the midst of data sharing agreement and getting access to the database that is being used. And the funding that we received last year will help in those processes as well.
We're going to move to the Board of Animal Health. I would like the DNR to follow up with Representative Becker-Finn and other members who've been having questions. So thank you. Dr. Beth Thompson, Board of Animal Health.
Mr. Chair, can you hear me okay?
Yes, welcome and introduce yourself for the record.
Thank you, Mr. Chair, and please let me know if we have a condensed time here, Mr. Chair. Thank you for the opportunity to give a status update on the board cervid program. My name is Beth Thompson, and I serve as the Executive Director of the Board of Animal Health, and I'm also the state veterinarian. I've got along with me today Dr. Linda Glazer, who is the Assistant Director for the board and the Program Director for the cervid program. She'll have a few slides to share after I get done here, and I'm just going to take a few minutes, folks. I want to walk through very high level that 3-page handout that was supplied to all of you and add a few comments. And then I'll also turn that question for the cost of a registration of a farm-served farm also over to Dr. Glazer, as she is the program director. And very quickly too, I want to appreciate, say my appreciation and agree with Mr. Balzer's comments on the co-management, and also Mr. Myers, Assistant Commissioner Myers' comments on the co-management. I really appreciate all of the work that has been going on amongst our agency staff on implementing the concurrent authority. So again, there is a handout, a 3-page handout for everybody. The Board of Animal Health really wanted to have that information in your hands so you can walk away from this meeting and have that information. If there is any additional information that you would need, for example, on page 1, when we talk about 257 herds currently registered within the state of Minnesota and the species that are associated with those herds, if anybody would like to see those numbers over time, and I can tell you those numbers are decreasing over time, but if anybody is interested in that data, please let us know. The same is true with the number of CWD tests that we did in state fiscal year 21. And then also at the top of page 2, we have compliance activity for this program, which includes the type of violation, the number of violation, correction orders, and all of that information right down to the number of cases that we've referred over to the Department of Revenue for collection. What I'd really like to spend just a moment on, Mr. Chair, members, first of all, regarding rulemaking, that was included in this handout, last few paragraphs on page 3. To further expand on that information, the board had been in the process of rulemaking for the farm-served program since early 2020. The first request for comments opened in June of 2020, and then you guys all have probably heard this. We formed a rules advisory committee. We had sessions with the tribes and tribal organizations, and we had sessions with served farmers and the public. If any of you have any questions about that process, the meetings, the drafts of the rules, the advisory committee report, I'd ask that you visit our webpage. We have most of that information all up on our webpage. As we were wrapping up that initial draft, and again, the initial draft of the rules, it wasn't the final draft, it was the initial draft of the rules in spring of 2021, we began scheduling needed meetings for our citizen board, our 5-member board. So we sent out the public notice on June 18th, just a few months ago, to let folks know we were going to have a special meeting. Then on June 25th, we posted that special meeting date, which was July 28th, again, for the approval of the draft of the rules. Based, though, on the legislative change to the statute pertaining specifically to white-tailed deer, the board meeting was canceled. And we are now, again, working with DNR on white-tailed deer specific rules as part of that bigger package of the served program rules. Lastly, and there have been some questions about monies paid by DNR and the status of the fence in Beltrami County. First of all, the legal action that was taken with regard to that Beltrami County herd owner and carcass disposal site, the first step was the board worked on and secured an agreement between the state agencies and the county of Beltrami for access to that site. And I think it was one of the DNR folks that presented, explained that the carcasses were dumped on a county-owned site. Then secondly, the board issued what's called a remedial action order to the owner of the CWD positive Beltrami County herd. In that order, we are seeking to hold the farm-served producer responsible for the costs of the disposal of those quarantined CWD exposed farm-served carcasses on that land. So those carcasses were later determined to be from a CWD positive herd. That herd owner is entitled to a contested case proceeding. So he has requested that proceeding, and legal action is currently ongoing. So the board is working very closely with the Attorney General's office and of course our other state agency partners on this. This is an ongoing and contested legal matter. And with that, Mr. Chair, members, I would like to turn it over to Dr. Glazer because I think she has some needed information on the herd status, Mr. Chair.
Dr. Thompson, I'm going to quickly have Representative Acom and Representative Eklund ask their questions quickly so that maybe they can be incorporated in Dr. Glazer, your testimony, because we need to get that public testimony in. So Representative Acom and then Representative Eklund quickly.
Thank you, Mr. Chair. I'll be brief. My question is about the licenses and Dr. Thompson, I think you said over time they've been decreasing. I'd like to know specifically how many new licenses we've had each of the license, I guess, applications we've had each of the last 5 years to understand if any kind of moratorium that might, some areas of the state are proposing, if they might be effective in any way. And then also wanted to know how long the licenses last. And lastly, if there are any things that would automatically revoke a license holder's abilities to have a deer farm. So thank you.
Representative Eklund.
Thank you, Mr. Chair. And my question, Dr. Thompson, and maybe Dr. Glazer could answer this as well, is we heard from Dr. Larson about the cost of this, and we've heard all along since we've been discussing this CWD issue that we need a live test. Well, Dr. Thompson, we have a live test. I want to know whether the Board of Animal Health is going to recommend this live test for all cervid farms. Thank you, Mr. Chair.
Dr. Glazer and Dr. Thompson.
So Chair Hansen, Representative Eklund, and others, you are correct. The law changed this past year that the board shall allow movement of cervidae from a cervidae farm located within a CWD management or endemic area if the cervidae has been tested negative with an antemortem test validated by the USDA. In addition to that, Representative Eklund, the process that generally is in place for quote unquote new testing would be for whoever wants that testing to be done to come to one of our quarterly board meetings, get on our agenda, and let's talk about it. And certainly we would include, whether it's Dr. Larson from the research aspect, Dr. Torreson from the VDL, if that's where the testing is going to take place. But let's move it through that process. So to your question, yes, let's start utilizing the test that is out there. Thank you.
Dr. Glazer.
Good morning, Mr. Chair and members of the committee. My name is Dr. Linda Glazer. I'm an assistant director with the Board of Animal Health. And first I can answer the questions from the representative regarding the licensing fees, and then I'll talk to you a little bit more about the CWD investigation and the details of the herds that were quarantined as a follow-up to that investigation. Our licensing fees have a breakdown of $500 annually or $250 annually. And the amount of the fee charged to the farm cervid producers based on the operation and sort of the herd usage for that herd. So the categories that were enacted by the legislature for, financial consideration, exchange, barter, a business operation, those, or a herd with multiple species are charged $500 a year. And those herds that do not have that sort of business component or commercial component to their herds, basically they're for a hobby, they would be charged $250. And that's an annual registration fee or inspection fee, we call it. And every year at the beginning of the year, we invoice producers for that fee and collect it either through online payments or checks that are mailed in. And that funding will obviously continue to decrease as the number of herds continue to decrease in Minnesota. Is there a way, the other question that was asked was about, is there a way to cancel we call it canceling the registration. And so we do have herds where we cancel their registration, which means they can no longer legally possess farm cervidae in Minnesota. That was on the update of the handout that we gave to you, just a brief paragraph about how many cancellations were done this year and a follow-up from cancellations from last year. So we do We do cancellations. We've done, I think it's been 11 to 12 in the last 3 years. And we, with that cancellation, the producers can no longer possess farm cervidae and they must get rid of the deer, the cervids, and we work with them to accomplish that. And so I just wanted to provide you a little bit of detail. The slide I have up right now is a diagram related to our CWD investigation. In the center of that slide is the Beltrami County herd, which was found positive in May of this year. And as required, when we find CWD infection in a herd, we trace the animal movements in and out of those herds for the previous 5 years. And so this Beltrami County herd was recently assembled in the fall of 2017. The producer was building the herd, so had not moved any animals out of the herd in the history of this herd. So we didn't have that tracing component. But what we had to do is go back and look what animals were moved into that herd. What were the sources of those animals that were found CWD positive when the herd was depopulated? And of the 12 animals that were found positive, 3 of them were sourced from other herds in Minnesota. And if you look at that diagram, you can see the blue arrows pointing inward to the Beltrami County herd. Those were the herds that contributed animals that later were found CWD positive when the herd was depopulated. The Winona County herd, Kanabec County herd, and Hennepin County herd. Those herds were quarantined. And in addition, any animals that were housed with the animals on those herds, but had since been sold to other locations, we had to find those animals as well. They were considered CWD exposed animals. In other words, they resided with that animal that was later found CWD positive while it was in either the Winona, Kanabec, or Hennepin County herd. So as you can see, Winona had moved animals out, to a number of different herds. The Houston County herd identified as CWD infected last fall, as well as those 3 herds on the upper right-hand corner, Morrison County 2, Stearns, and Crow Wing County. The Kanabec County herd had moved animals out of its herd to Morrison County 1, Mower County, and Mille Lacs County, those 3 boxes on the lower right-hand side. And then the Hennepin County herd had moved animals to a Dakota County herd. So what happened when these exposed animals were identified is that we had the animals appraised by a federal appraiser and requested indemnity, or the owners, said they wanted to request indemnity for those exposed animals. And we did request indemnity from USDA, and were granted indemnity in 6 of those herds. And those are the Morrison County 2, Stearns County, Crow Wing County, and then Morrison County 1, Mower County, and Mille Lacs County. And so the exposed animals in those herds were put down and tested for chronic wasting disease, and no chronic wasting disease was detected in any of those herds. So at that point, the quarantines were released on those 6 herds. The Kanabec County producer did request indemnity, but at the initial appraisal and request, there was no funding available from USDA at that time to provide, because that was the largest herd involved in this investigation, and had about 100 animals that were exposed. So they did not receive any indemnity funding. So the 6 herds up in the upper left-hand corner, the 3 in the lower right-hand corner are now, their quarantines are released. There's 4 herds that remain quarantined, and my next slide will provide some more detail about that. So of the 10 herds that were quarantined in Minnesota, well, as far as the trace investigations, just to let you know, those producers also may have sold animals to other states. What we do in that situation is identify those exposed animals and the information about them, and forward that information to the other states to follow up on the investigation in that state. But in Minnesota,
Dr. Glaser, we're going to have to wrap up your testimony. If maybe you can go through the slides and then answer that question from Representative Acom of, have there been any new registrations of farms in the last few years?
Okay, so the 4 of the 10 herds remain quarantined. The Winona County herd has an appraisal scheduled. They recently requested an appraisal, had not done so before that. The Kanabec County exposed herd, we have information that we may be receiving money from USDA that will provide indemnity for this herd. And if that is, we get final approval of that, that herd will be depopulated in the near future. The Hennepin County exposed herd is elected to stay quarantined for the 5 years from when the positive animal last left the herd. And if so, that 5-year timeline will be reached next October of 2022. And then the Dakota County herd has not yet accepted indemnity for those appraised animals. And Mr. Chair and Representative, I don't have the exact numbers of new licenses, but every year we do have new producers that want to get into the farm survey business. It is typically under 10 herds in a year, but I can get that information for the past 5 years and for the specific number of herds if you're interested in that and get that to you.
Thank you, Dr. Glaser, and members and the public. The other handouts from the Board of Animal Health are posted on the website. We do have the filming crew has been very tolerant in giving us some extra time, and we have a little more time, and I want to make sure we get the commissioners and all the other testifiers. So if Commissioner Boyle and Commissioner Nelson maybe could hop on and give their presentation.
Representative Hansen, we're having trouble with our audio here, but I think you can still hear us, can you?
We can hear you, so if you want to proceed. Yep.
Well, thank you very much, Chair and committee members for having us. I'm happy. This is Commissioner Boyle with Commissioner Nelson. We co-authored a moratorium that passed this morning in Virginia, Minnesota. And it was on a 7-0 vote with 6 of us active deer hunters on the county board. The reason why we did this, the straw that broke the camel's back, is the testimony on what's happened with the passing of a contaminated cervid animal in southeastern Minnesota to Beltrami, and what has happened since to Beltrami County. St. Louis County is very unique in the aspect that we have just under 1 million acres of tax forfeited land. And with that, we have over 100 leased deer hunting shacks, which is very unique in Minnesota. That gives the opportunity for some folks that may not be able to purchase land to be able to hunt. That brings in just under south of $500,000 to offset the levy for St. Louis County. And it's been happening for decades and decades. Our hope is with this moratorium is that it starts a domino effect with the great help of what we've heard from Dr. Peter Larson, who's the nation's leading expert on prions from the University of Minnesota, and Director Craig Engwall from the Minnesota Deer Hunters Association, and his work he's putting in with his 20,000 members plus. We have to start the domino effect of other counties starting a moratorium on no new cervid farms. And we need, more importantly, the state to lead the charge. And that includes the transportation of cervid animals from one game farm to another, and that absolute freezing of any new cervid farms. So I'm going to hand it over to Commissioner Nelson for a few more thoughts.
Commissioner Nelson,
Thank you.
Introduce yourself for the record.
Commissioner Keith Nelson, St. Louis County 6th District. Thank you, Representative Hansen and others for hearing our testimony today. I think first of all, I'm a 40+ year deer hunter, but I'm also a beef farmer in St. Louis County, and I'm very, very concerned that we continue to allow the movement of animals that have shown exposure to CWD. This could have an impact on not only the industry around hunting, but also the proud tradition that we have here in St. Louis County of an annual event that is a family event. It's a family-oriented event. And I really think it's time for action, which is why we did the moratorium. And we're hoping that, again, others follow the lead. St. Louis County is the largest county, obviously, in the state of Minnesota, with the largest amount of public land in the state. And that exposure to our wild herd is a concern that I think all of my fellow commissioners and I share. So any work that can be done, whether it's through the Board of Animal Health, whether that's through the DNR, it is absolutely imperative that those two entities work together going into the future, as they would. I said to my colleague here that if this was a case of iones in a beef herd, the action would be swift. But yet CWD in a deer farm doesn't seem to bring the same level of concern. And I, quite frankly, don't understand it. I hope that the moratorium is something that can be supported not only here in St. Louis County, but statewide. And thank you for your time.
Thank you. And the testimony, there's written testimony provided, as well as some of the public testimony, and the resolution from St. Louis County is available on the website. Next up, Chairman of the Fond du Lac Band of the Lake Superior Chippewa, Kevin Dupuis. Welcome, Chairman Dupuis. Chairman Dupuis, or is it Vice Chair Savage?
Mr. Chair, why don't we just move on? They said they might.
Yes, there's written testimony from Fond du Lac and from the 1854 Treaty Authority as well. We'll go to Brad Gausman with the Minnesota Conservation Federation and the CWD Action Coalition. Welcome, Mr. Gausman, if you could introduce yourself for the record.
Absolutely. My name is Brad Gausman, Executive Director of Minnesota Conservation Federation. Thank you, Chair Hansen and members of the committee for meeting to discuss this very important issue. As I said, my name is Brad Gausman. I represent the Minnesota Conservation Federation. Since our organization's formation in 1936, we've been involved in many important conservation initiatives. We were instrumental in the creation of the state's first hunter safety courses. We were involved in the passage of the Legacy Amendment, and we currently introduce new participants to hunting, fishing, and trapping through our Conservation Leadership Corps program. Our tradition of advocating for the woods, waters, and wildlife of our state continues on with the work I am speaking of today. My purpose here this morning is to address the threat that chronic wasting disease presents to our state's wild cervids on behalf of the Minnesota Conservation Federation and the CWD Action Coalition. The CWD Action Coalition is represented by the groups you see signed on to our resolution document. These groups represent not only the statewide deer hunting community, but also science-based groups, national conservation partners, and tribal nation interests. CWD is presently moving across our state and becoming more prevalent on our landscape with each passing year. This always fatal disease has the potential to wreak havoc on our state's wild cervids and have impacts far beyond the death of one cervid animal. Without a doubt, the captive cervid industry has proven to be a major vector for the spread of CWD across our state. The presence of CWD on the landscape in Minnesota can be traced back to the movement of captive cervids for the benefit of a relatively small private industry. Now is the time to enact a moratorium on any new cervid farms in the state, engage in a buyout of this industry, and to enact a ban on the movement of live captive cervids and bodily fluids originating from captive cervid animals. Thank you for your time and consideration of the document our coalition has provided. the CWD Action Coalition, and Minnesota Conservation Federation will continue to advocate for the health of our state's wild cervids and look forward to engaging with the Minnesota legislature to accomplish the goals set forth in our position statement. Thank you.
Thank you. The testimony is provided with the coalition on the committee website. Next up, Matt Lee, Backcountry Hunters and Anglers. Mr. Lee, if you could introduce yourself for the record.
Absolutely. My name is Matt Lee. I'm a Minnesota resident, and I'm the current chair of the Minnesota chapter of Backcountry Hunters and Anglers. Chairman Hansen, members of the committee, thank you for this time and opportunity. We gathered a lot of information today. A lot of what the BAH had to say in their handouts was missed. Unfortunately, that's a lot of important information as well. Please, I encourage everyone to check that out that hasn't. It's been 20 years we've been dealing with this on the landscape in Minnesota. We still don't really have an actionable plan. I represent 1,600 hunters across the state of Minnesota, and my organization also represents over 40,000 nationally. This is a pervasive issue that we need to do something about. We fully support the CWD Action Coalition's desire to have a moratorium on new farms, as well as shutting down the current operations, if we can't come up with an executable plan. We're robbing hunters to pay for these operations, right? The operations that are acting in bad faith. And unfortunately, the difference in dollars spent, $500 million spent in Minnesota in deer and deer hunting related activities. By the best estimates of the cervid industry, $25 million in 2016, I believe it was. That's 20 times more money spent by Minnesota hunters than anything involved with cervid farming in this state. We ask that this committee make actionable moves and have legislation ready to go when this new session opens in the spring. So thank you for this time and opportunity again. Thank you everyone for staying late, and I look forward to working with you on this issue in the future. Thank you.
Thank you.
Mr. Chair, could you try calling on Mr. Savage again? I think he was trying to unmute.
Okay. If Vice Chair Savage with the Fond du Lac ban is available. We'll give you a moment here.
Can you hear me?
Yes, we can. Welcome.
Yeah, this is Bruce Savage. I'm the District 2 representative for the Fond du Lac band. I also serve as the vice chairman. The chairman couldn't make it today. And we represent 4,200 band members who have been hunting deer in the Minnesota region since 1854 with our treaty affirmed rights. CWD impacts us at a level that it's hard for the state to even understand. But not only does it affect our cultural impacts and our historic and traditions, but it also affects our economy, which is a very fragile economy within reservation communities. And I would really like the state to take this very seriously. Now, what effects the CWD has on the lives and traditions of the Fond du Lac band members, and also the rest of the Minnesota Chippewa tribe, which represents over 40,000 members within the state of Minnesota. So when you bring this to legislation, please remember that our reservations are very fragile with the economy that our members can continue with, that we've done for generations. And to save a few of these deer farms and their income is a minor fraction of what the impact it will have on 40,000 members of the Minnesota Chippewa tribe. Thank you very much for your time. That's all I have to say.
Thank you. I want to make sure I praise Minnesota House Info for giving us the flexibility to go over significantly on our time, but we will not be able to go over much more. Members, we will have a follow-up hearing just prior to the firearms season to go over some of these same questions and updates to make sure we have a little more information. There is a quarterly report required that we probably would be hearing around that time as well. Members, I think there is a lot of information, and there was information referenced that is available on the website. I would encourage members and Minnesotans to read all of that information, particularly the Board of Animal Health enforcement activities. It was testified today that we have spent over $14 million in Minnesota public money, whether it's hunter money, whether it's general fund money, $14 million on this. And we're going to spend a lot more. It's just kind of incomprehensible that we're at this point. We have been dealing with this actively with votes in the Minnesota House for 5 years. And there are things that we are going to have to do. I'm very concerned that co-management, how that will work, if it will work. It may just be ongoing the same track we are on. So I appreciate the public testimony, the questions. I think there are going to be more questions, and we will have a follow-up hearing in late October before the next season. So with that, we are adjourned.